Let's see what happens. Okay, guys. Thank you for coming. There's a lot of you here who are not normally here. Thank you for taking the time out of your day. I appreciate it. I was like, what am I missing? Is there an exam? So here we are. So I'm hoping to finish everything for signal transduction today. And what we finish today is what will be on the exam. Quick business. Today is open recitation, meaning it's optional. Come if you want. Come if you're a Thursday person and you can come anytime between 1 and 5 today. Come today because you guys will not have that opportunity on Thursday. If you are desperate to meet with me anyways, I can make some time on Friday probably, but don't. I could try to make time for you on Friday or Monday. So just try to come when you can. We're going to just be doing practice problems. Actually, you're just doing whatever you want, really, and I'm there to help you. So there's no lecture or recitation on Thursday. You guys are so lucky. You get 2 weeks in a row with only one class. You get more time to study, right? It's great. Exam 2 is next week. I'm going to change it up so that you can only do your multiple choice first and then the short answer, so that way we can cut down on the typing sound. So we're going to try to figure out how to make that happen in a way that's efficient. We've got 2 hours, so maybe what we'll do is you'll do in the first hour you'll do one part and then the second hour. I don't know. We'll see. There's an option for oral exam. A number of people have been talking to me about it. I have yet to schedule anything. The oral exam will be next Wednesday for whoever wants to do it. So email me if you want to get on the schedule. Exam questions are due by 5 p.m. Friday. Last time I got a whole lot of recall questions, and that's why they weren't used. Make sure they're not recall questions. If you're saying, if it's something that can be a definition, that's a recall question. It needs to be something where you have to actually think, and use the information. So it's hard to write a good question, so keep trying. Okay, so take 5 minutes to write. I know this is a lot, but I just wanted to make sure you guys were thinking about RAS. So RAS plays a role in spreading the signal from one receptor to multiple pathways, right? That's what it does. So what is RAS? How is it regulated? Meaning what turns it on, what turns it off? What do you know about that? How would you expect mutant RAS to be found in cancer cells? What conformation would it be in? What kind of activity would it have? If you had a fluorescently labeled antibody to RAS and you used it to stain your cells, where would you find fluorescence? Would fluorescence change in the presence of ligands? Would it change if RAS was mutated? So spend 5 minutes thinking and writing, and then we'll go over it. Okay. How are we doing? Do you guys want another minute? No, it's a lot to answer. One more minute. Okay. Okay, so Amanda Maxwell. Okay, so what is REST?
So I said RAS is a molecular switch regulated by GEFs and GAPs. I said when it's GTP bound, it's usually on, and GDP bound is off. I said in cancer cells, I would expect RAS gap to be inhibited by some mutation that would cause RAS to always be on, because usually cancer results from gene overexpression, from what I've learned in other classes. Where I expected to find RAS, I wasn't sure. I was thinking maybe if it was, I think maybe outside of the nucleus, like, and then if it's bound by a ligand, it would be inside the nucleus, I think. And then it would change if it was mutated, but it depends on the mutation.
Okay.
So it depends what's being affected there.
Okay, so you are, that's perfect answer for the first part. How is RAS regulated? Yes, by it's a molecular switch, GTP bound, GDP bound, GAPs and GAPs, all of that. Everything you've learned about G proteins and GTP binding proteins, that all applies to RAS. So that's all right. How would you expect mutant RAS to be found in cancer cells? So that has multiple correct answers. One of which was yours. That's right. You said it would be maybe there would be some mutation that prevented the activity of a gap, so you would have no GTP hydrolysis, so it would be kept on. Therefore, GTP bound. What's another mutation someone else maybe thought of? Yeah. Something in the RAS gap. Would that keep it on or off? That would keep it off. Would you expect RAS to be is inhibition of RAS signaling in line with the idea of uncontrolled gene expression in cancer? Maybe in some cases, depending on the signaling, but mostly we think of, and so it could be something as simple as an interaction with the gap, so you're not hydrolyzing GTP. I guess that's probably the best answer. Yeah, that's my favorite answer. If you had a fluorescently labeled antibody to RAS and you used it to stain your cells, where do you expect to find fluorescence? So the reason I put this in is because I want you guys to think about where RAS is acting in your cells. And so it's a GTP binding protein. Does it ever go into the nucleus? My guess is no. Signaling pathway. So I want you to think about signaling from top down, right? So you've got your receptor, and when your receptor binds to ligand, you get recruitment of your RAS GAF. And that's what activates RAS. So that means RAS needs to be somewhere near the receptor. And so if you've looked at the figures in the book or in my lectures, you'll see it's actually bound to the membrane. So I wanted you guys to kind of pick up, because where things are, if you can kind of think of things as where they are in the big picture of the pathway, it's kind of easier to think about all the different pieces. So you would expect to find the fluorescence around the membrane. Did anyone get that? Okay, a few people did. Good. Do you think fluorescence would change at all in the presence of ligand? No, I see people nodding no. Because RAS is always in the same place. What regulates its activity is recruitment of the GAF. Would it change if RAS was mutated? I guess there are multiple answers to that one. Who wants to share their answer?
I said it's a possibility if you mutated the transmembrane domain, maybe it's not in the plasma membrane anymore, but at that point you find it outside of the cell.
Oh, okay. So if it's, I guess it depends on how. Yeah, you're probably right. So that was really good. Okay. Let me back it up. Right. So Austin said if the transmembrane domain was mutated, you might find it secreted from the cell. And I was going to say, well, yeah, I guess it depends on how it's bound to the membrane. If it has a transmembrane, I believe it might be. I almost feel like it's not GPI anchored, but anchored to the membrane in a different way. But either way, if you inhibit how it's interacting with the membrane through mutation, it should be secreted from the cell. Because one would guess, it depends on how it gets to that membrane, but if it's following the secretory pathway, then that would be secreted out of the cell. But if it's not following the secretory pathway and it's binding some other way, then maybe it would just be cytoplasmic. So that's where I'm like, I don't know actually. Because if you look at the figures in the book, it shows like a red squiggle, that membrane domain, but I'm not 100% sure, so now I have to go look that up. But very good thinking though in bringing things from different parts of the class together. But so if it was mutated in terms of binding to your gap, would it change the localization at all? No, right. Okay, good. Okay, very good, guys. Maybe this will work. Oh, is it? Well, that's good. It was just slow. Okay. So far, this is what we've done. We're slowly coming to an end. It's examples of receptors and the types of pathways that activate, right? We've talked about different, and so the way to think about it is from top down, right? So if you start with your signaling molecule, think about, is it hydrophobic, is it hydrophilic, is it some receptor bound to another cell? What kind of receptors have we spoken about that these different signaling molecules are going to interact with? We spoke about nuclear receptors, so those are our hydrophobic molecules. We've spoken about ion channel coupled receptors, but really only in the context of releasing calcium from the ER after GPCR activation. We spoke about GPCRs in fairly in depth, and enzyme coupled receptors. We've been talking about mechanisms for turning off signaling, and we're going to finish that bit up today. We spoke about how GPCRs work, how we turn them off. That's the arrestin part. We spoke about enzymes that are activated by GPCRs, adenyl cyclase and phospholipase C-beta, and second messengers that are released, and the example pathways that they activate, top to the bottom, right? We spoke about enzyme-coupled receptors and how they're shut off, and RAS and MAPK and scaffold proteins. And then we spoke about the different domains that are associated with signaling. Mostly we spent a lot of time with SH2 domains that bind to phosphotyrosines, right? So when you get all that phosphorylation of your receptors with your enzyme-coupled receptors, that's usually creating docking sites where signaling proteins that contain SH2 domains are going to be recruited. And we kind of used that a little bit in practice problems where we kind of thought about how we might experimentally determine if receptors are involved with signaling. Okay, so let's continue with our receptors. Oh, that's interesting. Oh well, we'll try to fix that on the final thing. So RTKs, receptor tyrosine kinases, they activate RAS via adapters and gaps, just what we spoke about. And here's just an example in how this works in eye development in Drosophila. So Drosophila have a compound eye made up of 800 identical units, and they develop from epithelial cells that are recruited in a specific order. And this is right out of the textbook. It's explained very nicely, so if you don't understand this, go back, read the figure legends and the accompanying text, and it should make a lot of sense. But basically what it comes down to is it has to do with the order in which each cell is recruited. That's going to basically tell that cell to develop in a certain way. And so it'll start with the center cell, and it will recruit the other cells, and through interaction between a receptor and a ligand, which is actually a receptor on the neighboring cell, it'll activate the signaling that encourages the gene expression required for photoreceptor development. And so you have this receptor tyrosine kinase, which has this intrinsic kinase activity. And this would be in the R7 cell. So this is this cell that's being recruited. It's actually the last cell that's being recruited here. And so this R8 cell that's in the center here, it has a receptor, which is called BOS. Don't worry about the detail, but I guess it is bossing the other cells around. And so it's going to bind to the RTK. So that's receptor ligand interaction, activation of your RTK. So you're getting phosphorylation through the intrinsic kinase activity of the RTK, and then a SH2 domain containing protein is going to be recruited, which in this case is an adapter protein that recruits a RAS gap. They all have different names: DARK, SOS, 7, there's all sorts of names, but if you just think broadly, you have a receptor that recruits the adapter protein, which recruits RAS-GAF, which then kicks off GDP, and then GTP can be combined and activate RAS, and then you have downstream signaling, with the whole point being activation of gene expression that determines the cell fate. Like you have a cell that has it's not differentiated yet, and just through interaction with the neighboring cell, it's being told turn into an ommatidia. And that's going to be one of those compound eyes. So, so interaction between the two different cells is determining gene expression for development of photoreceptors. So RAS activation is very quick, right? So you don't want to keep something that's stimulating multiple different signaling pathways on for a long time. So it's activated quickly, and then it's quickly reversed by phosphatases. So this was determined experimentally using a method called FRET, which is fluorescence resonance energy transfer. And so basically what you're doing is looking at a change in fluorescence to understand whether two proteins or two biological molecules are interacting. And so what they had is they had RAS that was engineered to be fused to YFP, yellow fluorescent protein, so it's fluorescent yellow. And by being fluorescent yellow, it's emitting a certain amount of energy. And then they have this GTP that is red. And so when RAS is activated, what is it bound to, GTP or GDP? Right. And so when the GDP is kicked off and GTP is bound, then this red dye is close to the YFP, so it transfers the energy and makes the red fluorescence. So then red appears. So this red dye, you can't see it all until it's activated through the energy coming from the YFP. And so that's why it's called fluorescence resonance energy transfer, because it's the transfer of the energy from one fluorescence to the other that makes the second one bright. And so what they did is they added a signaling molecule, in this case EGF, and they just looked at how long you see active RAS for. And how do you know, how were they able to detect if RAS was active? What color was it? Orange? Red, yeah. Right, red because this GTP would be bound and it would be bright. And so by seeing, you add your signaling molecule and see the red appear, and then the red's going to go away when GTP is hydrolyzed. And so then you can see it's really within about, I don't know, 4 minutes or so that RAS is turned off. So it's very quick and brief. Any questions about this? This is a pretty cool, I always love these kinds of experiments because it's a quick color change. It's either there or it's not, right? So it's either active or it's not. Okay, so I've been talking a whole lot about signaling and how activation of signaling often results in gene expression, but then I keep saying, but it's not, that's not the only thing that signaling activates. There's other things too, like cell shape and cell movement and stuff like that. But the most obvious and easy one for me to explain and understand that I think students understand very easily is gene expression, right? You have a virus, you need to respond by mounting an immune response, and cytokines are going to bind to receptors, activate gene expression, and then you fight the virus and everyone's happy. But also, I don't want you guys to think that's all that happens, so we're going to just do this quick example here of how signaling affects cell shape and movement. So the F receptor is involved in the guidance of an axon tip to the muscle target. So if you want to have those connections, you need to have the cell growing in the right direction, so it's in close proximity to the muscle cell, so that you can have movement, right? But not all. You don't always want every axon going to every muscle cell, right? You want only things going in the right direction. And so here you're having negative regulation from signaling. So you've got your F receptor, which is an RTK, right? So it has its own kinase. It has its own kinase activity, intrinsic kinase activity. And then you're going to get recruitment of another kinase. But really the big part to pay attention to here is effexin, which is a rogue GAF. So automatically, GAF will get another GTP binding protein. And so what rogue GAF does is it activates Rho. And you're going to say to me, well, what's Rho? And so this is the introduction to the next unit that we're going to be doing, starting next week, about the cytoskeleton. And how the cell, the shape of the cell, the movement of the cell, is very much regulated by actin and the building and the breakdown of actin in the places where it's needed. That makes sense a little bit now, but it's going to make a lot more sense as we start to learn about that. You can imagine actin, if you're growing your actin, so your actin filament is growing and it's shooting out in this direction up against the plasma membrane, it's going to force the cell to move in that direction. And that's kind of how cell movement works in a very generalized way. So what's happening here is if you have a nearby cell which has a receptor that's going to bind to the F receptor, what it's going to do, it's going to activate effexin, which is this Rho GAF, and then you're going to get activation of Rho A, and that's going to tell the actin to form in a certain way that's associated with contraction that will cause the growth of the axon tip to collapse. So it actually won't grow. So it's negative regulation. So in the absence of a signal, when you want the forward movement of your axon towards the muscle target, you have effects in there, but it's active different. It's active at a lower level where it's activating all these different um, row family members equally. So you have growth point extension. So you have your axon growing in the right direction. But when you have epherin binding, so that's the ligand, it's going to phosphorylate and activate effexin in such a way that it only activates row A, which is associated with contraction. So rather than moving forward, it's contracting and collapsing. So then you don't have that growth. And it kind of keeps the cell movement going in the right direction by getting rid of the ones that shouldn't be going in that direction. So just keep this in mind as we move towards cytoskeleton, it will make more and more sense. Yeah? Huh? Yeah, so this is this epherin A1. So it's cell communication. That's the signal. Questions? So I just want you guys to understand it's not only gene expression, that we can regulate these proteins that regulate actin polymerization and cell shape and cell movement through signaling as well. Okay. So PI3 kinase, it's a kinase, right? And it can be activated by multiple receptors. So this is an intracellular signaling molecule that can be activated by RTKs, GPCRs, it can be activated by RAS, but it's a kinase that phosphorylates inositol phospholipids. So this should kind of make you think, oh wait, GPCR is associated with something like that. And it creates docking sites for intracellular signaling molecules. And so it's another signaling molecule that helps to spread the signal downwards. So remember with the GPCRs, we had phospholipase C beta that got activated, and that cleaved off the phosphate groups, and that cleaved, so you end up with diacylglycerol and IP3. IP3 went and activated the calcium channel, and ER opens it up. Flood of calcium, activate KC, all that diacylglycerol became a docking site, right? So that was what GPCRs were doing, but then also you can have PI3 kinase, which is this kinase that will phosphorylate these phospholipids and create docking sites for recruitment of other proteins. And then obviously wherever you have a kinase, you're going to have a phosphatase that can also remove those phosphate groups. So this is kind of the same story as we've been saying all along. It's all about phosphorylating something, creating a docking site, and what you're doing is you're recruiting more signaling molecules that are going to spread the signal down the pathway. P10 is the phosphatase that removes this phosphorylation and removes those docking sites, so it's just shutting off the pathway. And it's actually frequently mutated in cancer. What do you think the outcome is? Yeah, you have overexpression of genes because you can't shut off the pathway. Because phosphatases are going to remove those docking sites and get rid of those signaling molecules and turn off the signaling pathway. And so if you've got your phosphatase not working, then you can't turn off your signaling pathway. Do you have a question? P10 is a phosphatase, exactly. So PI3 kinase is involved in cell growth and survival. So again, if you have an issue turning off pathways that are involved in cell growth and survival, then you're really going to have a problem because cancer is when you have too much cell growth and survival, right? Kinase. So you'll have some sort of signaling molecule that's going to bind to some sort of receptor tyrosine kinase. So again, this is another example of a enzyme link receptor that has intrinsic kinase activity, has a kinase domain. So when the ligand binds, you get the phosphorylation of the receptor. makes a docking site for PI3 kinase. And then PI3 kinase is going to phosphorylate these inositol phospholipids and create docking sites. See, there's nothing docking, and now there's docking sites for these other proteins. AKT and PDK1, don't worry about the details, but remember, other proteins, most of which are all kinases, are recruited then to that inner side of the plasma membrane, where then it can be, the AKT can be activated, and now that kinase can go on and phosphorylate things that are going to have an effect on something. In this case, you get phosphorylation of BAD, which is an inhibitor of an apoptosis inhibitory protein. So once BAD goes away, then you have this apoptosis inhibitory protein. That's what cell survival is, right? Inhibition of apoptosis. So suddenly you've got this protein that inhibits apoptosis that's very active, inhibiting apoptosis, right? You want apoptosis if you're trying to prevent cancer, right? But when you suddenly have so, of all these proteins here, a lot of them you could imagine you find mutated in different cancers, right? So you might find issues with AKT a lot of the time. AKT is a big one. PTEN, which was the phosphatase, that's going to remove these phosphates. Remember we talked about PTEN? Phosphatase removes those phosphates, so there's no docking site. And so that kind of cuts off the pathway right here, right? So, yeah, so that's an example, signaling through a receptor tyrosine kinase, phosphorylation of phospholipids, creating docking sites, recruiting other kinases, activating those kinases. It's all the same. And then effect on some effector. Okay. Questions about this? No? Okay. And so that's not complicated enough. Often there's crosstalk between different pathways. Signaling through a GPCR and signaling through a receptor tyrosine kinases sometimes will activate the same pathway. Do not memorize this, please. I just want you guys to understand that it's complicated. And so there's never one, so I give you a lot of examples just so you can have some concepts of how things work. But if you go and memorize all these pathways, it's not useful because there still might be different things. So. Okay, so we talked about enzyme-linked receptors that have intrinsic kinase activity. They have a kinase domain in the receptor. Then we've also got kinase-associated receptors. So these are receptors that don't have intrinsic kinase activity, but they bind to a kinase. Outcome's the same, right? You have a kinase somewhere that's going to phosphorylate something, but it's just where is that kinase coming from? Is it a different protein, or is it within the receptor itself? Okay. So just to, what is wrong with this today? Something happened. I was just going through all these slides in my office, working on them, and none of them were all wacky like this. I'm going to blame you. No. It's really annoying. So we already learned that SH2 domains bind to phosphatase. SH3 domains bind to a proline-rich sequence. I promise I will fix these if they're funny on Blackboard. Yeah?
Yeah.
Well, that's good. That's really annoying. I hate computers. Here's again. Jack Stapp, something's probably wrong with my PowerPoint. I bet you it's going to crash in like 2 minutes. Yeah. They bind to proline-rich sequences in other proteins that contain this PXXP motif. So basically it's a protein-protein interaction domain. Who's raising their hand? No? Okay. Marking crazy. So that's supposed to say Jack stat pathway. Apparently it's just the stat pathway. So we already went through this one in the first lecture, but now we're going to revisit it now that you know all the brilliant things that you know. So Jack's stat pathway is an example of signaling through a tyrosine kinase associated receptor, right? Because you've got these Jack kinases that are associated with the receptor. And when you have ligand binding, that brings the receptors close together, and then you can get activation of the kinases by phosphorylating each other, which then they phosphorylate the receptor, create a docking site. Again, same theme, a bunch of phosphorylation, create a docking site for something. In this case, STAT. So you get recruitment of STAT proteins, and then they get phosphorylated, which contains binding sites so they can dimerize and bind to each other. And only when they're dimerized can they go and enter the nucleus. And so this is a gene regulatory protein that's inactive except for when it's a dimer, and then it can enter the nucleus. And so how is signaling turned off here? So we just turned it on, right? This is actually an antiviral response. So you have a virus, all these poor Ebola patients. Hopefully you're doing a little of this right now. Who's scared of Ebola? No, don't be scared. Be scared of the flu. Did you guys get your flu shot? I haven't got mine yet either, but it's on my list. Huh? It hurts this year?
Yeah, like everyone that I got it
Maybe the person administering it
was
was bad. Anyways, so once you have your signaling molecule binding to your receptor, you get activation of your antiviral response. Do you want that always on? No, that's really bad to be mounting an immune response when there's no virus. You need to turn it off. So how would you turn this off? Who has a hypothesis of how you might turn this off? You don't need to know the answer, you just need to think about it. Yep. Right, so endocytosis of the receptor, that's one thing. What else? Dephosphorylation, so phosphatase can remove these phosphate groups. Yeah. So those are two things that you can do. Right, so phosphatases, they either modify or turn off the signal. And the reason I say modify or turn off is because just like phosphorylation doesn't always turn something on, it's really just creating a docking site to recruit something. Phosphatases don't always turn something off. They're just removing that docking site. So depending on what's being recruited, if you're recruiting an inhibitor, you're not turning something on normally, right? So if you're getting rid of an inhibitor, you're not turning something off. So you always have to be careful when you say turn this off, turn this on. But if you say a phosphatase is going to remove a phosphate group and that protein can no longer bind, so this outcome's going to happen, that's usually going to be right. So obviously you're always going to need a little more information about a specific pathway before you can say whether or not a phosphatase is going to turn something on. Phosphatases, they can be cytoplasmic or plasma membrane bound. This is just showing how phosphatases, so basically it's showing there are these similar domains of phosphatases. So here, these are these membrane bound phosphatases, but it's not really known what they're doing. Maybe a little bit more is known now since the textbook was written. But notice they all have this red box, and these are just depicting, they all have tyrosine phosphatase domains. So within the protein, they have a bit of it that has the activity of being able to remove a phosphate group from a tyrosine. That's how they're all similar. That's why they're all tyrosine phosphatases. They all have that same structure. Notice this one here also has SH2 domains. What do those do again? They bind to phosphotyroxines. Right, so if you're a phosphatase, would you want to be recruited to your phosphotyrosine? Maybe. Otherwise, how are you going to get native substrate? Things to think about. Okay, so I just want to introduce the concept of latent gene regulatory proteins. We've kind of been talking about them, but I guess I haven't called them that very much. So these are proteins that activate gene expression, but that's what a gene regulatory protein is, right? It regulates gene expression. A latent gene regulatory protein is one of those proteins that's inactive until it's activated. So when it's latent, it's not active, and when it's activated, it's active. Sometimes I say things and I'm like, why did I just say that? I said something the other day, and I was like, it's a global world we live in. My husband's like, is that a Hopkins PhD talking? And I'm like, yes, it is. Hopkins PhD is a global world. Then my daughter said, I'm dying a death. And I was like, oh, I know, it is a global world. It's okay to be dumb sometimes. So we've been talking about this, and I'll go through a few different examples. But basically, they're excluded. These are proteins that are gene regulatory proteins, and whether or not they're active really depends on whether or not if they're in the cytoplasm or the nucleus. If you're a gene regulatory protein, your job is to bind to some region near the promoter or an enhancer region just upstream of the start site for transcription on the DNA and recruit RNA components. That's your job as a gene regulatory protein. If you're not in the nucleus, can you do that? You can make me die a death. Right, if you're not in the nucleus, can you bind to the DNA if you're in eukaryotic cell? No, forget, I don't pay attention to bacteria, so I don't even know what they are. Sorry, guys. No, if you're a eukaryotic cell and you're not in the nucleus, you cannot activate gene expression. I would say we could pretty much agree on that. So latent gene regulatory proteins, some examples that we've talked about are like in the JAK-STAT pathway, what you see is that dimerization of STAT. The STATs are always there. They're just not in the correct conformation for them to be able to enter the nucleus and bind to the DNA and activate gene expression. And so that's why they're latent gene regulatory proteins. They're inactive. And they're excluded from the nucleus, and they're off until some sort of signal through a receptor activates them. That's signal transduction. So the TNF and NF-κB pathway, that's another example of a latent gene regulatory protein. You always have your NF-κB, that's your gene regulatory protein. It's just normally bound by an inhibitor. So it's inactive. But when you have signaling, the inhibitor is degraded, and that allows you to enter the nucleus. We went through that once before. I'll go through it one more time. And then SMADs are another example. So I'll go through these. But that's a general concept to understand latent gene regulatory proteins and how they're regulated. They could be regulated through phosphorylation, dimerization, conformational change, degradation of an inhibitor, all these different things that can happen that will allow them to go from the cytoplasm into the nucleus and bind to the DNA. So SMADs, they are latent gene regulatory proteins, and they are activated through TGF-beta signaling. So again, you've got a receptor. This is a receptor that has its own intrinsic kinase activity. One ligand binds, come together, phosphorylate each other. And what happens, so you get activation of your kinase, right? So it phosphorylates this kinase, which then gets activated. Then it can phosphorylate SMAD. And so SMAD is there, but it's just in a different conformation. It's in this inactive conformation. And when it's phosphorylated, it changes its conformation. So it's not folded up, it's open. I mean, that's how it looks. And so once it is open, it can interact with SMAD4. So SMAD2 and SMAD3 represent latent gene regulatory proteins. They're present in the cytoplasm. They're unable to enter the nucleus until they're they dimerize. Oh my god, this whole thing is all crazy looking. I'm so annoying. And then they can enter the nucleus, activate gene expression. Questions? As long as they're not questions about my crazy slide. I'm worried about the lack of questions. Let's have a heart to heart here. Is this really simple for you to have questions, or are you completely lost?
I guess I have a question. So when you have the phosphorylation of the SMAD2 or SMAD3, it's just coming up to the kinase and getting phosphorylated, and then it's going down to doing another one, dimerize.
Yeah, so basically, exactly, your SMADs are in close proximity to your kinase, so they can be phosphorylated, and then it's available for interaction with SMAD4. Hi. Yes.
So all of these two regular proteins are they occupy the pathway normal or not normal?
So the question is, are all latent gene regulatory proteins up by the plasma membrane if they're not in the nucleus? No, they're just in the cytoplasm. Sometimes they might be up by the plasma membrane. It depends on the protein. But just think of cytoplasmic or nuclear. Any other questions? Thanks, guys, for fighting the good fight there and coming up with some questions. Okay, so here's a quick practice problem, and we're not going to spend too long going through it because I want to make sure we get through the rest of this stuff. Who even knows what it says? Phosphorylation of SMAD induces interaction with SMAD4, of SMAD2/3 with SMAD4, and translocation to the nucleus. What assumptions could you make about SMAD2/3 and/or 4 in terms of nuclear localization? So right now, what you need to do is think to yourself, okay, so when you phosphorylate SMAD2/3, there's some kind of conformational change that allows interaction with SMAD4, which also changes its conformation. And then it can enter the nucleus. And so now you have to remember what you know about nuclear localization and nuclear import and what proteins need to interact with for nuclear import to happen and what you think might be happening here. Yeah, right. Yeah, it's probably like STAT where it doesn't have an exposed nuclear localization signal until the conformation changes and you get that randomization. Right? The nuclear localization signals bind to importance and bring proteins into the nucleus. Right? Were you going to say the same thing? So that's good. So you were taking so that's exactly what I want you guys to be doing with all these examples that I give you, is try to look at them and say, well, how are these similar? How are they different? How is this an example? What can I take from this and apply to something else? Okay, because on the exam I might say signaling pathway gorilla and signaling protein mouse do this, and you're not going to be able to be like, oh, I memorized that because it doesn't fit. So you're just going to have to think about what you know. Yeah.
I think I have a question. The SMAD2 is that phosphorylating SMAD4?
So look at the proteins here. So the way to know that in the textbook, when there's kinase activity, usually they make those little red hash marks, and then you see a phosphate group added. And so you know that these are phosphorylated. This one's phosphorylated by the receptor, but SMAD4 actually never gets phosphorylated.
Oh, okay.
And SMAD2/3 don't have kinase activity. It's just the fact that the phosphorylation changes the conformation of SMAD2/3. Well, and then if SMAD2/3 is in this open conformation, it can bind to SMAD4 and change the conformation. Anyone have questions about this practice problem? Okay, so signaling pathways. Okay, so remember, we talked about the receptor. We talked about some of the intermediate signaling molecules. And now we're talking about these effector proteins that need to get into the nucleus to activate gene expression. And so this is really about regulation of latent gene regulatory proteins. And so what we saw with the SMADs is just a conformational change, right? There's also another way to regulate latent gene regulatory proteins is through regulated degradation of something, proteolysis. Right, so I'm going to give you 3 examples here. Notch, Wnt, and NF-kappa B. Wnt you already know because we did. I think we've seen Wnt and NF-kappa B multiple times already, and we did the paper and recitation on Wnt, so it's definitely useful to understand that pathway. Notch, this will be probably the first time you're seeing it, but they're all similar in that something's degraded or cleaved in order to activate the pathway. And so you've got a latent gene regulatory protein that's inactive in the cytoplasm, and then becomes changed in some way to something being cleaved or degraded so that it can go enter the nucleus and activate gene expression. So you want to compare these when you're looking at them. So Notch signaling is involved in what's called lateral inhibition in development. And so this basically means one cell telling neighboring cells not to follow its fate. Okay, so communication between cells. And so here again, you don't have a soluble molecule as a ligand. It's a receptor as another cell surface molecule that's going to bind to the receptor. So you've got Notch, and then you've got delta. And so the way it works is you've got your Notch receptor, and as soon as it interacts with delta on the other cell, you get cleavage of the cytoplasmic tail. And that cytoplasmic tail is the latent gene regulatory protein. And it is able to then migrate to the nucleus and then interact with other gene regulatory proteins and activate Notch target genes. So here you've got cleavage of the cytoplasmic tail. Wnt, I don't even really need to go over that because you guys know it so well, but I'll do it just for fun. So with Wnt, you get binding to your receptors. So actually, in the absence of Wnt, you have this constant degradation of beta-catenin because beta-catenin is being phosphorylated by GSK3, and then it's being targeted for degradation. So this latent gene regulatory protein is regulated by constantly being degraded. And then when you have Wnt signal, it recruits GSK3 to the receptor, and then it gets sequestered in multivesicular bodies, and that allows beta-catenin to be stable. That's how that latent gene regulatory protein is controlled. And so then it can go into the nucleus and activate gene expression. And so you actually have GSK3 being incorporated into multivesicular enzymes, so it's unable to phosphorylate beta-catenin. And then that allows beta-catenin to go into the nucleus, bind to DNA, activate gene expression. And so then there's the NF-kappa B pathway, where you've got, again, all the same, ligand binds to receptor, activation of kinases, and then what happens is this kinase phosphorylates I-kappa B, which is an inhibitor of NF-kappa B. And so here your latent gene regulatory protein is NF-kappa B, but normally it's inhibited because it's bound to an inhibitor. And phosphorylation of its inhibitors targets it for degradation. And so then NF-kappa B can go into the nucleus. And so in this case, what's happening is your inhibitor is binding to your NLS. So you're not able, so NF-kappa B is kept out of the nucleus because you cannot see, the NLS cannot be seen by the import. Questions about this? No? It's because you guys love NF-kappa B, right? That's probably my favorite pathway, I'd say. Okay. So just quick wrap up here. When you're starting to think about studying, start categorizing things, right? GPCRs, they signal through G proteins, they regulate signaling through effective small mediators, the second messengers such as cyclic AMP and calcium. Understand how that is different from signaling through these enzyme link receptors, either RTKs, receptor kinases, which have this intrinsic kinase activity, or the kinase-associated receptors, like in the JAK-STAT pathway, where both of those types of receptors are pretty much just activating other kinases, which somehow affect an effector protein. And then as we move down the signaling pathway, we can start thinking about regulated proteolysis and the activation of latent gene regulatory proteins. And then you want to compare and contrast these latent gene regulatory proteins and how they're activated. Are they activated through phosphorylation and a conformational change, or are they activated through degradation of something, like what we just went through? Oh, good. Horrible. I say we just throw this computer out the window. Oh, that's probably why they don't have windows in these lectures. Because the faculty would be like, so these are just thoughts and concepts as you're studying. I really do not want you guys to be memorizing the laundry list full of pathways. That's not useful. I want you to be thinking to yourself, you know, what activates the signaling pathway? How is this regulated? Why is this regulated? What's the end goal? Right? Big picture, and then you can actually think about it. What would be the outcome if this became dysregulated? How could things become unregulated? Each pathway you should be able to look at and be like, okay, well, if this didn't work right, it might not work. If this didn't work right, something might go wrong. Always remember, signaling turns on, but signaling needs to turn off as well, right? And so these are mostly things that we've talked about already, right? You can get sequestration in multivesicular bodies. You can get degradation in lysosomes. You can have a receptor be inactivated through arrestin, for example, or through recruitment of an inhibitor. You can have intracellular signaling molecules be inactivated. You can get production of an inhibitory protein. So when you're thinking about turning off signaling, I want you to think of it in two different ways. One major mechanism is inhibition at the receptor level. And don't ever tell me take away the ligand, because that's not how the cell turns off signaling, right? That's how the researcher might, or the whole overall system might turn off signaling, but I want you to think within the cell, okay? So one is inhibit signaling at the receptor level through these different mechanisms. Then the second major mechanism is through inhibition of signaling intermediates. What are signaling intermediates? What does that mean to you? Something between the receptor and the outcome. Right, so something between the receptor and the effector protein. Any of these kinases. Anytime you turn, you have to think of it as like, it's a pathway, you know, there's a start and there's an end. Anytime you inhibit something in the middle, you can shut off the pathway. And so, two different mechanisms. One, stop something at the receptor level through maybe degradation of the receptor. And the other is inhibition either through dephosphorylation of an intermediate, degradation of an intermediate. So we talked about endocytosis as a mechanism for desensitization with beta-arrestin. And then we spoke about endocytosis and degradation. We spoke about endocytosis and recycling. So sometimes you might just need to downregulate for a short while and then bring it back. We spoke about sequestration and MVBs. We spoke about turning off GPCRs either through GTP hydrolysis or through beta-arrestin and endocytosis. And so then also these intermediate signaling molecules. So for example, the JAK-STAT pathway can be inactivated through two different mechanisms, right? You can get degradation or inactivation, right? So degradation is where you actually take one component of the signaling pathway, induce ubiquitination, and then degradation of that protein. If it's not there, it can't participate in signaling, right? Or you can get dephosphorylation. So you can get degradation of kinases, inhibition of kinases,